In this class we will learn about transplantations. So I will talk about the basic concept of transplantation and how scientists come to know that there are involvement of immune system during transplantation. So the first is, what is transplantation? Transplantation is nothing but donation of organ or tissue from the donor to the recipient. And during this transplantation there are two outcomes occur. That is, acceptance of the tissue or the organ or rejection of the tissue or organ by the recipient. And in the beginning I want to mention here four type of transplantation can occur depending on the donor: autograft, isograft, allograft, xenograft. Depending on the donor, there are four type of transplantation can occur. The first is autograft. That means tissue are transferred from one site to the another site of the same individual. So autograft means same individual. Transplantation occurs same individual. Like in a burnt tissue, burnt individual, tissue skin can be transferred from one side to the burnt site. Isograft means when donation of the organ or tissue occur between genetically identical individual. This genetically identical individual can be strain of mice. This is called inbred strain of mice, or identical twin in case of human or other animals, that also known as syngenic. The next is allograft. That means genetically different individual, and these two different individual that is genetically different are called allogenic. Actually from same species. The last is xenograft. That means from different species. In general it has been seen that autograft and isograft are generally accepted, but this allograft and xenograft generally not accepted. That means they are rejected. The rate of rejection actually depends on the tissue. Like skin is rejected faster than heart and kidney. So now I will show you three experiments by which scientists have come to know that there are involvement of immune system during transplantation rejection. In the first experiment, scientists took two different strains of mice. They transfer skin graft from strain A mice to the strain B mice, and they found that the strain B mice reject, because these two mice are genetically different. That means this transplantation is actually allogenic. The strain B mice reject the skin graft after approximately 12 to 14 days. They call it first set rejection. After a few days, scientists another time skin grafted from strain A to the strain B, and they found that now the strain B mice also reject the skin, but it takes 5 to 6 days. So they call it, this is the second set rejection. So during the second set rejections, the time is somehow low than the first set rejections. So scientists come to know that there is some memory. Memory is actually generating in the strain B mice towards the strain A mice skin graft. So they are generating some memory. They perform another experiment, very simple. They took another strain, let's suppose strain C, and grafted skin from the strain C mice, and they found that the strain B mice reject the skin graft from the strain C and took approximately 12 to 14 days. That means this mice is showing the first set rejection. So now they perform a very important experiment that is called adoptive transfer. They isolate T cell. T cells means CD4 cell and CD8 cell, both cell, and transfer this cell to a strain B mice. So this strain B and this strain B mice are actually syngenic. That means they are genetically identical. Not only that, this strain B mice are actually unprimed. Unprimed means this strain B mice, these mice have not experienced any grafting from strain A. So this is called unprimed. When the second transplantation occurred, that means this mice is actually primed, because it has actually experienced the first set of grafting. But this mice is not experienced any grafting from strain A. So this is called unprimed mice. They actually inject this T cell from the strain B mice to this strain B mice that is actually unprimed. This is called adoptive transfer. Scientists took the strain A mice and grafted skin strain A to the strain B, and they found this unprimed, unexperienced strain B mice showing rejection after approximately 5 to 6 days. Scientists actually found that when first time tissue transplantation occurred, because this is the first time this mice is experiencing this skin grafting, and they found from this first experiment that when first time tissue transplantation occurred, the mice take actually 12 to 14 days to reject, that is the first set rejections. But here, as the scientist actually injected this T cell from this experienced mice in the unexperienced mice, this mice is actually not showing the first set rejections, this mice is actually showing the second set rejections. And scientists come to know that this CD4 and CD8 cells are actually somehow responsible for the rejection of tissue transplantation or organ transplantation. That there is another strain of mice that is called nude mice. In this mice there is a mutation in a protein, and for that reason these mice don't have any hair, so they are hairless, so this is called nude mice, and they don't have thymus, or some mice have vestigial thymus, so they don't produce CD4 CD8 cells, they don't have cell mediated immunity. Okay, that means CD8 or CD4 cell mediated immunity. These mice do not reject allograft and also xenograft. Now you can understand what is the role of this CD4 and CD8 cells. So those CD4 and CD8 cells actually helps in the rejections are called, in case of allograft, they are called alloreactive T cells. In the second experiment, this is very simple experiment, scientists perform three things. First is they remove CD8 positive cells from the recipient mice to see whether there is any role of CD8 cell, and they found that those mice don't have CD8 cell, they reject the allograft or xenograft and take approximately 15 days. So now you can see there is no change when there is no CD8 cells. After removal of CD8 cells, there is no change in the rejection period, because I have told you in normal case the first set rejection requires 12 to 14 days, and in this case after removal of CD8 cell the mice is showing the same rejection period, it takes approximately 15 days. Now scientists perform another thing, they remove CD4 positive cells, and they want to know what happens now. After removal of CD4 cells, they found now the tissue rejection is actually slowed down. The period actually 30 days. So the mice take 30 days to reject. Okay, so there is a very important role of CD4 cells. They also perform another thing, another experiment. They remove CD4 positive and CD8 positive both cells, and they found that the rejection period now prolonged approximately 60 days. So from these three data they come into conclusions that there is correlation in between CD4 positive and CD8 positive cells, because when they remove only CD4 cells it takes 30 days to reject, but when they isolate, remove both the CD4 and CD8 cells, they found that now the mice reject the tissue and take approximately 60 days. So now tissue rejection actually prolonged, the time requires to reject the tissue prolonged. So there is some correlation between CD4 and CD8 cells. Now I will show you another experiment by which they come to know, scientists understand that there is a molecule that actually very important for this tissue transplantation or transplantation rejections. This is the third experiment. Scientists coming to know a very important molecule that is actually responsible for tissue transplantation rejections. They call this molecule major histocompatibility complex. The name implies major. Major means it has a major role. In histo means tissue, compatibility means compatible, or you can say that the recipient, whether a recipient is compatible for the donor tissue or not. and complex means the molecule. They took different strain of mice, a strain that have MHC molecule H2B, and they took another strain of mice that have H2K MHC molecule, and they crossed these two strain of mice and found F1 generation that carrying H2B and K, not B or K, B and K, means both MHC molecules. Now they perform very simple experiment I am showing you. They took the H2B mice and transplant tissue from H2B to H2B strain, H2K strain, and also to the F1 generation H2B or K. They performed simple another experiment. They took H2K mice and transplanted tissue from H2K to H2B, H2K, and also the F1 generation, that is H2BK. And they also performed another experiments. They took H2BK strain and transplanted H2BK mice, H2B mice and H2K mice. Tissue transplantation occur between this H2B and H2B mice. That means same MHC molecule, those having same MHC molecule, tissue transplantation accepted. In case of F1 generation mice that have H2B and K, both MHC molecule, when tissue transplantation occur between this H2B and H2BK, tissue transplantation actually accepted. Only where there is a difference between MHC molecule like H2B and H2K, tissue transplantation is rejected. The same is true for H2K mice. When H2K mice tissue is transplanted to H2B, this is rejected. But as these H2K mice have MHC, same MHC molecule, they are accepted. These mice also have K, so they accept. And in case of, now this is the very important and very interesting fact, H2BK mice, when tissue transplantation occur between the same strain H2BK, tissue transplantation accepted. But this mice is generating from this parental strain, because you can see H2B is the parent, H2K is the also parent. So these are the parental strain. But when this F1 generation mice is transplanting tissue from this H2BK to H2B, the tissue is not actually accepted, it is rejected. Also in case of these mice, this is rejected. So parental strain could not carry the F1 generation mice. Why? Because you can see here, these mice have H2B MHC, these mice have H2B, so there is a similarity. But as these mice have H2K, a different MHC molecule that is not present in this case, in these mice, so this is actually not accepted. So here from this experiment, scientists come to know that similarity between MHC molecules is very important. So nowadays also MHC matching actually occur. This is called tissue typing. When tissue or organ is actually transplanted from donor to recipient, the recipient gene or the MHC gene are actually sequenced to match with the donor. This is called tissue typing. If there is any difference, then the tissue can be rejected. And if there is similarity, most of them that actually found that tissue are accepted. Now I will show you 3 important molecules. For that reason, very fast tissue rejection occur. Very fast. The first molecule is ABO blood group antigen. If there is any mismatch between ABO blood group, as we know there are 2 antigen, that A antigen, B antigen. So if there is any mismatch between this ABO blood group, then tissue transplantation rejection occur. And now you can ask me that why this ABO blood group is important, because we know that during the blood transfusions, when someone donates blood, then ABO blood group matching is important. But why MHC molecule is important for the transplantation of other tissue, like it could be heart or kidney or skin? The important fact is this ABO antigen is actually not only present on RBC, it is present RBC, but not only RBC, it can be present in the epithelia and also endothelial cells. So as the tissue have endothelial and epithelial cells, so this antigen can be actually, if there is any difference, the tissue can be rejected. The second molecule is major histocompatibility complex. The third is minor histocompatibility antigen. One of the example is HY antigen of male individual. All male have a antigen that is called HY antigen that is produced by Y chromosome that do not present in female. So this type of difference that is very subtle, it's called minor histocompatibility antigen. There are different loci present for this minor histocompatibility antigen. So these three molecules are actually responsible for the first rejection of tissue, and these are the experiment by which scientists come to know that there is involvement of T cells, and they found that CD4 and CD8 cells collaborate each other, and there is a role of MHC molecules. There are three types of rejection can occur. One is hyperacute rejections, acute rejections, and chronic rejections. And scientists have discovered many suppressive drugs that could suppress our immune system not to reject the tissue transplantation.